Segal, who's also from the Weizmann Institute.
Hi, can you hear me? Okay.
Yes, and you need to switch to the presenter mode for your presentation.
Yes, share screen, right?
Yeah, we can see your screen, or someone's screen. I'm not sure who's it is.
Maybe I need to stop sharing.
Oh, yeah, okay.
Oh, yeah, sorry. Yeah, right.
Okay. Let's see.
Yes, now we can see your screen in the right mode.
Good. Yeah, so hi, everybody, and thanks for the opportunity to present our work. We've been the past decade or so been focusing on working on various human cohorts towards personalized medicine with emphasis on nutrition and gut microbiome. And I'd like to tell you about a few of the projects that we've been working on. I guess the first large-scale project that we did was published several years ago, so I'll just go over it briefly, was when we wanted to understand what healthy nutrition is. And towards that goal, we initiated what we called the Personalized Nutrition Project, where we recruited 1,000 people, and we connected each of them to a continuous glucose monitor to check their glucose levels as they recorded all the meals that they ate. And overall, we had 1,000 people and about 50,000 meals, and this was published. So I'll just summarize the key results, which were, we showed on a scale of 1,000 people that people have very different blood glucose response to food, very reproducible within a person, as you can see in two curves for the same person eating the same meal on 2 different days, but very different across people. And we were then able to take personalized data that we collected on people, gut microbiome data, and various clinical features, and be able to develop an algorithm that quite accurately predicted these blood glucose responses, these personalized responses. And in that study, we also did a short-term 2-week intervention where we gave people with prediabetes who normally have huge spikes in their blood glucose levels, as you can see here. We gave them a menu designed by the algorithm with identical calories, and we're able to fully balance their blood glucose levels. And so this we published several years ago, and that begged the question of what would happen if people then adhere to such a diet for a long-term period, and whether that can have really clinical benefits. So we took on ourselves this challenge, and we recently completed 3-year work in the lab of doing a proper randomized clinical trial, where we took 200 people with prediabetes and randomized them either to either a standard of care dietary arm, pretty much the Mediterranean diet, or our algorithm diet. And since these were people with prediabetes, we looked as the primary outcome at the changes, the average change in glucose levels, so measured either by hemoglobin A1C or by a CGM, continuous glucose monitor, that we put on people for the entire duration of the intervention, which lasted for a period of half a year. This is anecdotally one participant connected for 1 month to a CGM prior to the intervention. You can see a lot of spikes in glucose levels. And during the first month of intervention, the diet pretty much eliminates these spikes in glucose levels. And for the entire half a year of intervention, we can see major reductions in this one participant. Here's another participant. On the upper left are the glucose levels prior to the intervention, and then the 6 months of intervention, where you can see a reduction in the spikes. And here's another participant, and another one. Overall, when we look and compare all participants, you can see comparing the red and the green prior to the intervention and during the intervention on the algorithm diet, huge advantage, huge reductions. And in contrast, here's a participant on the standard of care arm. The upper left panel is the diet prior to the intervention, and the The other panels are during the intervention. It's hard to see a difference. And here's another participant, and this is now summarizing the statistical results of the study across all 200 participants, 100 in each arm. On the left are the changes in hemoglobin A1C in the 6 months of intervention and then 6 months of follow-up. You can see a significant reduction in the standard of care arm, but then a reversal back to baseline levels in the follow-up. And that's compared to, we were very happy to see, a double the effect on the algorithm diet on hemoglobin A1C, and that effect is maintained in the 6 months of follow-up. And if we look here on the far right, are the changes in the spikes, the time above 140 milligrams per deciliter, and we see that the standard of care diet reduces it by about 30%, the algorithm diet by about 70%. So these outcomes we also believe are clinically significant because the NIH did a pivotal study called the Diabetes Prevention Program, showing that a even smaller reduction in hemoglobin A1C was able to reduce conversion of prediabetes to diabetes by about 60%. So we're hopeful that this bigger reduction can have even bigger impact. And this is an intervention that purposely was only a dietary intervention compared to the NIH intervention, which also included a caloric restriction and enhancement of physical activity. So, so this whole body of work for us completed a journey of about 9 years from an idea to development of an algorithm and then to an actual RCP, concluding with what we believe is a successful intervention that can be of therapeutic value for people with prediabetes. And we also have preliminary data showing advantages of this on people with diabetes. So very satisfying for us. Another work that we recently published is on the same cohort, examining a different aspect, which is looking at metabolites that circulate in the bloodstream and asking a very fundamental question, which is what determines levels of different metabolites that circulate in our bloodstream. And this question is of major importance because we know that metabolites that are in circulation and reach the body systemically can have impact on disease. So we'd like to understand which factors determine levels of these metabolites, because if we then have metabolites that are causative to disease, then we might have means by which we can then intervene, be it in the diet or in the bacterial composition, and by that modulate predictably levels of disease-causing metabolites. So for that, we measured over 1,200 metabolites using mass spectrometry. These are very common metabolites. Over 1,100 of them are found in 50% of the patients. They cover a wide range of different metabolites, including lipids, amino acids, some unknown metabolites, xenobiotics, and so on. And as means of trying to see whether we can predict these, whether we can understand the determinants, we thought that we'll use a machine learning approach to show that we can predict these metabolites out of sample. So the way this approach would work is, say that we wanted to predict levels of caffeine, which is one of the 1,200 metabolites that we measured, using dietary data. So we would build this matrix of dietary data on our participants and the measured level of caffeine. We would learn a function that maps the dietary data to levels of caffeine, a machine learning algorithm. We use gradient boosting decision trees. And once we learn that, we apply it to held out subjects using only their dietary data. We make a prediction and we compare it to the actual measurement. So standard machine learning approach. And when we do this, we can see that for caffeine, we can actually predict with very good levels its levels. But of course, caffeine is just one of the 1,200 metabolites that we measure, and diet is only one of the inputs. And so we built for each of the 1,200 metabolites, we built an algorithm using either one of the different input categories: diet, microbiome data, gender, time of day, anthropometrics, age, and drug intake, and so on. And the first question that we asked is how many of these metabolites could we actually predict by any of these input categories? And it turns out that for over 90% of these metabolites, we could get significant predictions, so meaning that our models on held out data could achieve a significant R-squared explanation of these data. The extent to which we could make these predictions, of course, varied from one metabolite to another. And when we look overall, we see that diet by far predicted the largest number of different metabolites, but in second place, the microbiome composition predicted several hundreds of different metabolites. And we then wanted to see whether we could replicate these and demonstrate to ourselves that these predictions were actually robust. So we collaborated with the group of Tim Spector from the UK, who also collected a similarly sized data set of coupled metagenomic and metabolite data. And we were very happy to see that when Tim sent us the metagenomic data and we never saw the metabolite data, we generated predictions, sent it to him. We saw that for the top 50 metabolites that were predicted in our Israeli cohort, all of these predictions also generated significant predictive power in the UK cohort. And actually, even then, all of the predictions that we made with significance, so 85% of them achieved significant predictive power. So that gave us confidence that these predictions are actually fairly robust, even given the microbiome composition differences between Israel and the UK. So, so these predictions are nice, but of course, we'd like to also understand what's driving them, namely, when we say diet predicts metabolites or microbiome predicts metabolites, which dietary components are dominating, which specific bacteria are dominating. And towards that, we used a feature attribution framework called SHAP, which is a nice framework that basically breaks down the composition, the contribution of different features in a machine learning model, taking into account the other contributions of all other features. So basically, it tells you how you arrived at a prediction. And what's nice is that the breakdown is additive. So you get the contribution of each feature, even though your model is non-additive, like non-linear, like the gradient boosting decision trees. And so using that, we can go back to, say, our model for predicting caffeine levels from diet, and we can ask which component is the most predictive. And here, not surprisingly, we find that self-reported drinking of coffee is the most predictive feature. So that's a good positive control where we know what we expect. And similarly, some other metabolites that are known either to be a marker for citrus fruit consumption or for fish consumption, the best driving feature was self-reported eating of either oranges or fish. So that gives us some positive controls and some confidence in the method. And we can now go to our microbiome predictions, like 4 metabolites predicted well by the microbiome that you can see here. And now we can ask, okay, which specific bacteria are driving these predictions, and we get specific species. Interestingly, the 2 metabolites on the bottom are unknown metabolites, and we now identify some species that may be driving these predictions. And the reason why this is important is because if you had reason to believe that some of these metabolites are positive, then probably the way to modulate them would be to intervene in these specific bacteria. So that's a step towards microbiome-based therapeutics, we believe. But everything I showed you, of course, on observational data, it's just correlations. So we wanted to ask, as proof of principle, whether we can show that at least some of these associations are causal. So we went to the diet where it's easier to do interventions. We took some of the predictions from sourdough bread, where we had some metabolites predicted to either be negatively or positively associated with sourdough bread. We recruited 20 participants. We gave them sourdough bread for 1 week. And indeed, we saw that metabolites that on the big cohort showed positive association with sourdough bread, they tended on average to increase following 1 week of sourdough bread intervention. So meaning we have a way to predictively intervene and increase these metabolites levels, which we predicted would happen based on the model. And this did not happen when we gave the same participants white bread, showing the specificity of these predictions, and again, proof of principle that at least in some cases, these associations may be causal. Diving deeper into the microbiome, an area where we have a big focus on, and where we are developing new ways to look and analyze the microbiome, which in my view will be the next frontier in microbiome research, which is to go down to the level of not just the bacterial composition, but really to the single nucleotide level of different bacteria. So similar to what we do in human genetics, where we do genome-wide association studies, now we're going to do metagenome-wide association studies. And the idea is that when we have metagenomic samples, typically today we map them to a reference database and we look at bacterial composition. But of course, when we do that, we have a lot more information, because we actually have the mapping of the reads, so we can really call the base pairs that we have for each at each bacterial genome within each sample, and we can then study this variation, either for looking at associations to human traits, for looking at transmission and evolution of bacteria within the gut. So our first focus and some results that I want to show you today are these associations that we have between single nucleotide changes in bacteria and various human traits. So the reason I think why this type of study was not done before is data availability. You really need a lot of data for this, especially in microbiome data, because most people will not have all bacteria, and when you even have the bacteria, you need to cover the positions in order to do these associations. And microbiome data has really been lagging behind genetic data, which we now have available on even millions of people. And comparatively, microbiome data is still in its infancy and available typically on hundreds sometimes, but very rare still on thousands. So we had access through a collaboration with a microbiome consumer company to 30,000 samples, about 27,000 from Israel, 4,000 from the US, which we used as a validation cohort. And so we did a standard association study, but this time on metagenomic data. There's some differences that one has to take into account when doing this on metagenomics that I don't have time to go into, but this is the result of associating about 75 million SNPs on 3,000 different species, and 60,000 of them come up as significant even when you do a stringent von Ferroni correction. And you can see the Manhattan plot here, where the y-axis shows the p-value of the association, some very significant p-values, but I think even more interesting than the p-values are actually the sizes of the effects shown here by the volcano plot for the association of with BMI. And you can see that there's quite a few SNPs that have a lot of different points of BMI that they associate with. And here's an example of one of these SNPs. So this is a single SNP in one individual bacteria. And you can see here that the individuals with the major allele, 18,000 of them, have on average a BMI of about 27, compared to the 500 individuals that have the minor allele, or a mix of the major and minor allele. They have an average BMI of 31. So these 3 or 4 points difference in BMI, that's a lot. That could translate to 10 or 15 kilograms of body weight. So the point is that the effect of these is very big, and I think there's a lot of opportunity to go much more deeper into mechanism, because now the associations are not just on levels of composition of bacteria, but really on changes and variation in individual genes. And these results also replicate in the US discovery cohort. The other point I want to bring up is, and give an example of one of these associations in BMI, is that unlike human GWAS, where typically every gene is represented in just one instance in the genome, in bacteria we have a lot of orthologs for the same gene, so we can look at variation in that gene again and again and again when it appears in different people, and we see variation in the same gene but in different orthologs. So for BMI, for example, a lot of the associations tended to be in one of the genes, the beta-galactosidase gene, which breaks down complex carbohydrates. And so an example to show you how maybe this variation that we see here is pointing us to potential mechanisms. I think in the interest of time, I'll skip over this, but we also have many significant associations with age, for example, that also explain quite a lot of years of age, and we can summarize. So far, we've done it only for gender, age, hemoglobin A1C, and BMI. We find many different associations, and we can look at the different pathways and genes where variation in them is associated with the phenotype. So in the final few minutes, I want to tell you about also some additional unpublished work where we've actually used interventions in the microbiome to see if we can treat some disease, and specifically atopic dermatitis. So atopic dermatitis, or AD, is a chronic skin disease that's quite prevalent in about 20% of children, about 10% of adults. It's a multifactorial disease. And so we attempted to do a fecal microbiome transplant in only 9 patients. So just to see if, as proof of concept, if there would be an effect. And the way the experiment was designed is that each person was his own control, and we give it by capsules separated by 2 weeks. Initially 2 doses of placebo, followed then by 4 doses of fecal material from a healthy donor. And I'll first show you the clinical outcome because I think it's quite striking. Here's a participant at week 4, so after 2 doses of placebo, and you can see the clinical manifestations of disease. But then after 2 doses of the FMT, you can see visually even remission and clearance of the clinical symptoms. And here's another participant where even at week 33, so long after the last FMT dose was given, we also see significant remission. And when the clinicians quantify this by a score of atopic dermatitis, what they call a SCORAD, you can see that after the placebo, there's no change. And there's then a 70% reduction on average in these 9 patients in the SCORAD. And the clinicians say that this is better than any known treatment that they have for this disease. So we obviously wanted to see whether we can identify under the hood what's going on in the microbiome. And so for that, we first looked at the baseline samples, and we looked to see if we can see transfer of bacteria. And so the way we do this is we put all the species here on the y-axis, and for each species, we compute all the pairwise distances between unrelated individuals. That's the x-axis here, showing on average, depending on the species, about a 99% or 99.9% similarity of SNPs across the entire genome. The dots here represent comparison of the species between the donor and the patient. And because these are baseline samples, of course, there's still no connection, no transfer of bacteria. And so the dots sit right within the average distribution of pairwise distances between unrelated individuals. And the same happens when we do a placebo. So again, we're not really doing any intervention, so there's no change in the dots. But when we actually do the real FMT, we see a lot of the dots shift to essentially a 0 difference, meaning the genomes are identical between the donor and the patient. And so we believe that this is with 100% transfer of bacteria from the donor to the patient. And we can see which bacteria these are. So, for example, this Prevotella copri, in 7 out of 8 patients that we analyzed, it was transferred from a healthy donor to the patient, and we can see some bacteria that transferred in more than one individual. And we're now expanding the study, and the idea would be to try and isolate some of these bacteria and see if a cocktail of them could actually be grown as a therapeutic for AD. And the final slide I'll show is just a global view of the microbiome. So we had 3 healthy donors here. All their samples are enclosed by ellipses here. And you can see that they cluster together, and the different patients are in different regions of this space. And after we do the placebo, there's really no change in the patients. But after the FMT is given, we see that patients are really shifting their microbiome dramatically, and they become closer, not identical, but much closer to their donor. So this is another way to kind of globally look at all the changes that we see here in the microbiome. So I think I am out of time, and so with that, I will skip to the acknowledgement slide of many different collaborators that we have, and all of the remarkable and amazing students that I have, and that really have driven all of these projects. And thanks for listening. I'll be happy to take any questions.
Thank you very much for the great talk, Eran. So I will start with the first question here. So for this first project you were talking about with a personalized diet, so I was just curious, do you think there is a way to sort of, because it was based on the glucose monitoring, right, that you needed to have for a month. I mean, is there a way to sort of say do like an approximate version of that algorithm that wouldn't need as much input, and then get sort of, I mean, because the results were very impressive. So even if you shaved off a few percentage points, it might be worth sort of that approximation to sort of improve over the Mediterranean diets. Have you tried that?
So yeah, so actually the approach does not require the CGM. The CGM was only for the monitoring. The CGM we used on the big cohort of 1,000 people, where we learned the algorithm. And once we learned it, when we have a new person coming in, we only measure his clinical data, personal data, and the microbiome. We apply the algorithm. So it doesn't require the CGM. The CGM was just for us, for monitoring during the study, to just look at the outcomes. So
okay,
I think it actually probably can be even further improved on a personal manner if you also had CGM data for a person. But I think, as you said, I think even we're happy with these results, and yeah.
Okay, so we have a question from Hannah Jacobs here. Do you take into account the relative amounts of certain bacterial SNPs in each patient when doing the meta-GWAS?
The relative amounts
of each bacteria, or the different bacteria.
Yeah, right. No, so we do not, and purposely, because I believe that in abundance estimation of bacteria, there's a lot of noise in that estimation, because that may depend. We saw it in our hands on collection methods and extraction methods and sample processing and so on. And it also depends, since these are relative abundances. It's always relative. It's never absolute. So there's that bias as well. Whereas when we look at the DNA we believe to be nearly 100% accurate. When you read a letter in a certain position, it's not an error. And so if we find associations between that, I think it's both more accurate and robust, and also it's kind of much more high resolution than just abundances of bacteria, which is why we're working on this approach. It brings us much closer, we believe, to actual mechanisms.
Right, thank you. So we have another question from Julian Liminatakis here, who's wondering about those remaining spikes in the glucose time series. So do you know what are responsible for that? Is that when people sort of deviate from their diet and eat too much chocolate, or what's the hypothesis there?
Yeah, so we actually have a log of people during the intervention, and yeah, I think again the algorithm is not 100% accurate, but it's fairly accurate, and I think a lot of that, yes, may be deviations, which, and you know, and that's fine. I think for half a year of intervention, if you overall you see a big clinical benefit, I think it's also absolutely fine that you also have some deviations and still you're improving your health compared to the situation you were at before.
Okay, thank you very much, Eran, for a great talk. And so we'll take a 10 minutes break here and be back after 2:35 here in the East Coast time for the last two talks. Thank you very much.